As head of the Department of Experimental and Clinical Pharmacology, it's my great honor to introduce our keynote speaker for today's Pharmacogenomics conference. An accomplished scientist, internationally recognized leader in the field of pharmacogenomics, distinguished professor of both pharmacy and medicine, and dean of the University of Florida College of Pharmacy in Gainesville, Dr. Julie Johnson serves as an exemplar for many of us who seek to advance translational medicine and research, moving forward from discovery to implementation, specifically in the field of pharmacogenomics. Julie received her baccalaureate degree in pharmacy from the Ohio State University, her PharmD from the University of Texas, and completed her postdoctoral fellowship in cardiovascular pharmacology and pharmacogenetics at Ohio State University. Dr. Johnson's research focuses on cardiovascular pharmacogenomics and genomic medicine. Her efforts in these areas have resulted in over 250 publications, peer-reviewed publications in over 40 million, an extra zero though, you're on your way, 40 million in research funding as a principal investigator. I know there were some heads that kind of raised there. This has actually earned her recognition, surprisingly and importantly, in the Thomson Reuters highly cited scientist category, representing the top 1% of scientists globally in the fields of pharmacology and toxicology. In addition to her scholarly achievements, she has served numerous roles in committees including the FDA, NIH, with leadership roles in ACCP, American Heart Association, and most recently stepping down as president of the American Society of Clinical Pharmacology and Therapeutics. Dr. Johnson was elected as a member of the National Academy of Medicine and has received numerous awards, including the Leon Goldberg Young Investigators Award, Paul Dawson Biotechnology Research Award, Therapeutics Frontiers Award from ACCP, and among others. In addition, she's been recognized as an excellent educator and by way of teaching awards received from both the universities of Tennessee and Florida. Julie's contribution to the profession of pharmacy and disciplines that engage translational medicine using tools such as pharmacogenomics is nothing short of extraordinary. We are all pleased to have her share her perspectives on improving clinical outcomes through pharmacogenomics. Please welcome Dr. Julie Johnson.
Thank you, Robert, for that kind introduction, and it's really a great honor to be here and share a little bit of the work that we have been doing. And you know, if I make only one point, I would like to argue that if we don't document improvement in clinical outcomes, pharmacogenomics will go nowhere. So I'll talk a little bit about the work we've been trying to do to accomplish just that. So I think precision medicine is a hot term. All of us, as was mentioned, is really at the forefront of trying to advance precision medicine. Precision medicine is not just, pharmacogenomics is not just genomics. It's a concept of using a variety of pieces of information about an individual, which may include where they live, do they have sidewalks, do they have a grocery store nearby to buy fresh fruit. But the reality is, at the moment, we are best able to use genetic information, and more specifically, to use genetic information in the context of drug therapy. So really, pharmacogenomics, while not the only piece of precision medicine, I would argue pharmacogenomics is the tool at the moment that we can best use to advance precision medicine. So this is a cartoon, and you already saw something very similar to this. Bill Evans and I published this in a review article in 2001 as our concept of the clinical potential of pharmacogenetics. We were a long way, in most cases, from clinical implementation of pharmacogenomics in 2001. But again, the idea is that we have historically treated people with a given disease as one group. We know there are at least three groups of people. So these are individuals who respond well to a given drug therapy, those who will not respond well to that therapy, and those who will have toxicity. The toxicity really comes broadly in one of two forms, one that's drug concentration related, so sort of pharmacokinetic driven, and the other that really is not related to pharmacokinetics. And so the concept is, what we do now most of the time is we give the drug, and then we say which of these three groups does the patient fall into, and we make a change from there if needed. And the goal is that we use genetic information within pharmacogenetics to predict that up front. So if they're orange and they're predicted to have a good response, all is good. If they're predicted to not respond well, and really the two examples I'll talk most about, we're really focusing in this respond well or not respond well, then we need to use a different drug. And if they're predicted to have toxicity, then if that's a kinetic-related effect, we may need to change the dose. If it's not related to kinetics, then we would, again, use different therapy. So the exciting thing is 17 years ago, Bill and I sort of put this out as a cartoon to talk about this concept, but we really are in an ability now to actually do this in some settings. So how do we get there? How do we achieve the goal of clinical implementation of pharmacogenomics? So first is the discovery of genetic variants that influence drug response. And there has been a lot of work, and many of those papers that were shown in that graph published in the last 15 to 20 years really fall into this category of discovery. Once we have discovered those genetic variants, we need to understand whether they explain enough of the variability to be predictive and therefore useful in the clinical setting. Not everything that's probably a true genetic association is going to be robust enough from a predictive level to be useful. So we have to develop tools to understand how to use those. And really CPIC, I think, in some ways is an important piece of these tools. Then we actually have to do it. We have to implement, we have to figure out, will healthcare providers, physicians in particular, actually use this? But then I would argue most importantly, once we implement, can we document improvement in clinical outcomes? So I could talk about any of these. I've spent a lot of the last 20 years, the first 15 focused on the first two pieces of that, but much more recently really focusing on the last two bullets. So I'll really focus today on the last two bullets, but I think it's important to recognize that we have to continue the discovery work as well. So we have been fortunate to be funded in an NIH network, NHGRI network that's called IGNITE, which stands for Implementing Genomics in Practice. And we're the only college of pharmacy funded in this network. There are 6 groups currently funded, and I have 3 of those highlighted in red. And they're highlighted in red because this is a genomic medicine network, not a pharmacogenomic network, but 3 of us are funded to do pharmacogenomics research. And the reality is that while University of Maryland and Mount Sinai are not funded to do pharmacogenomics work in this network, they have very active pharmacogenomics programs. So this is, again, part of my argument that, or part of my evidence for the argument that pharmacogenomics really is at the forefront. We have a website and toolbox for the network that we actually host currently at the University of Florida. And by toolbox, it's really just that. So lots of little pieces of information. So for those who are trying to start an implementation in the clinical practice setting, you don't have to start at zero. You're going to hear a lot later today about CPIC, but I just want to highlight how critical CPIC has been in really advancing this field. Internationally, this is really recognized as a consortium that has really helped to advance the field. And I hope I'm right that there are 34 guidelines. Kelly will correct me if that's not correct. Importantly, at the moment, they don't really focus on drugs that are guided based on somatic variation, so just germline variation, but lots of really great information and tools. And then just also to mention PharmGKB, which is really an extraordinary knowledge base for pharmacogenomic information, so if you're looking for that as well. So I want to really focus my time on 2 stories of pharmacogenetics implementation that we have done at UF Health, really focused on whether we can observe differences in outcomes. So one is the gene CYP2C19 and Clopidogrel, and then we'll talk about CYP2D6 and opioids. So just assuming that there's a few people in the room who don't know all about these genes. So CYP2C19 is an important drug metabolizing enzyme, has important genetic variability. And so STAR1 is typically what's considered the normal function allele. This is a gene that has common variation, common loss of function, or sort of null alleles, STAR2 being the most common among those, STAR3 also a little bit common. STAR17 is what we call a gain of function polymorphism. It leads to increased expression of the protein and then increased drug metabolism. So those combination, the genotype then that results, or the haplotype that results, leads to a predicted phenotype, a drug metabolism phenotype, which is either normal, intermediate, or poor metabolism, depending if you have 1 or 2 copies of a loss of function allele, and then 1 or 2 copies of the gain of function allele, rapid or ultra-rapid. So just to provide some context, the IM and PM is about 30% of the population, so about 30% of the population carries at least one loss-of-function allele, and about 30% of the population carries at least one gain-of-function allele. So it's only 40% of the population that actually has normal metabolism. So most of us are not normal, so when you pick up your results, you can see which group you fall in. So Clopidogrel, a very important antiplatelet drug, is an important part of this story of understanding how pharmacogenetics can influence. So Clopidogrel was improved in the mid-90s, was not fully recognized at the time that the compound was not actually active, that it was a prodrug, and so it's a two-step bioactivation process to lead to the active metabolite, and more recently recognized that CYP2C19 was an important enzyme in both of those steps. And so as I mentioned, the loss of function, or STAR2 allele in particular, is common. So about 30% of whites, as I mentioned, about 35% of blacks, and up to 70% of Asians carry a loss of function allele. So starting in the early 2000s, there was really a rapidly emerging literature documenting, and it's not hard probably for especially any pharmacist in the crowd, to recognize that if you have a reduced ability to metabolize via CYP2C19, you're going to get less active metabolite. If you get less active metabolite, you're going to have less efficacy, and that's been documented in terms of antiplatelet reactivity, so ex vivo tests of platelet reactivity, and then retrospective clinical trials with Clopidogrel documented that those individuals who carried a STAR2 allele were at increased risk of major adverse cardiovascular event, or MACE, following a percutaneous coronary intervention or balloon angioplasty and stent placement. So these are the CPIC guidelines for Clopidogrel, first published in 2011 and then updated in 2013. And they're really focused on patients undergoing PCI or acute coronary syndrome. If there are genotype data available, the STAR17 allele in this case does not seem to change what we would do in therapy, but the recommendations in CPIC is that those who carry one or two loss-of-function alleles, so IM or PM, should be prescribed alternative antiplatelet therapy. Currently, those choices are prasugrel and ticagrelor. So we implemented this at UF in almost exactly June 25th of 2012, so almost exactly 6 years ago, and we began testing patients in our cardiac catheterization lab, and this is just a screenshot of the webpage on the day that we launched. And so we really believe firmly, and I think all of us working actively in this field believe that one of the key elements to success is we cannot assume that the busy physician or other clinician is going to know all of this. So we have to tell them what to do with it. And so we have worked really hard in our electronic health record to build tools that help in this implementation. And so this is a screenshot. We only give an alert when we want them to do something different. So we never tell somebody, the genotype is good for Clopidogrel. It's fine. If they were to Clopidogrel and the genotype is good, they don't hear anything from us. But if they have a loss-of-function allele and an order is placed for Clopidogrel, it triggers this alert. And so we've tried to be very concise in how we present the information. It's yellow, so it tries to catch their attention. And so you see at the top we say, problem, this patient's genotype is associated with very impaired metabolic activation of Clopidogrel and elevated risk for stent thrombosis and other cardiovascular events after PCI. Reasons is sort of one clinical pharmacology statement about what's really going on, and then we have specific recommendations, prasugrel or ticagrelor. We also give them the contraindications and cautions for those two drugs, and then they can make a change. So in the first tab, they can cancel the Clopidogrel order and order prasugrel, same thing for ticagrelor, or continue. So I'll highlight two other things on this screenshot. First is that the genotype is there, but it's very, very small. We do not want them focused on the genotype. We want them focused on the decision making. Now, there is a lab report that has sort of all the typical lab report information that they can go look at, but we do not want them focused on, having to remember, oh yeah, STAR2 is a problem. You also see that there's a hyperlink there for more information. We haven't looked in a while, but the last time we did look, nobody had ever clicked it. And so this goes to the CPIC guidelines and tons of literature, but I think what this tells us is that if you work on educating them when you do the implementation, they don't care. They don't want to go back and read the primary literature. They sort of trust that you're the leader in the field and you're not going to guide them down a bad path. So like I said, we implemented starting in 2012. We published our data really on the implementation success over the first year. And so we had over 1,000 CYP2C19 tests reported into the electronic health record. Of those, about 300 had undergone a PCI. So we were doing everybody through the cath lab, not those just getting a PCI. So by the end of the year, 84% of those with a PCI had a test ordered. And you can see in the first 2 months versus the last 2 months of the year, we had increased uptake. When we looked at the actionable genotypes, for those who were poor metabolizers, 100% had their drug changed. And for intermediate metabolizers, 67% had drug therapy changed. So we're like, great, we had a successful clinical implementation. We did it, we got it into the system, the pathology lab developed the test, it got reported back, physicians made drug therapy changes, but we really said, do we really know it matters? Do we really change anything? So then at about the 2-year mark, we felt like we had enough data that we could go back and look at outcomes, and I haven't really quite figured out how to use a pointer, so just bear with me. So we're really showing 3 lines here. So the worst line, this is a survival curve, so you want to stay at 100% because you want to stay alive. This is actually alive without stroke or heart attack. So it's a composite of death, heart attack, and stroke. And so the dotted line that goes down to about 85% over the first couple of months are those individuals who carried a loss of function allele. That meant we would have recommended prasugrel or ticagrelor, but they continued on clopidogrel. So we didn't think that was a good idea, but for whatever reason, they didn't get switched, and they had significantly worse outcomes than those individuals who did get switched, which is the solid line, or those individuals who stayed on clopidogrel but did not carry a loss of function allele. So these were really exciting data. We showed significant differences in outcomes, important outcomes, death, stroke, heart attack. I don't think anybody would argue that those things don't matter. We presented those at the AHA, and everybody's like, that's great, but it's not a randomized controlled trial, and it's only UF, and maybe it's just a single center kind of thing. We're like, all right. That's great. So then within the IGNITE network, we began to work collaboratively and pulled together 7 academic medical centers in the US who had done similar implementations. And so we then had data on over 1,800 individuals, about 30%, which is very similar to what we saw at UF, about 30% carried the loss of function allele. And of those, again, all of those would have had alternative therapy recommended. About 60% had their drug therapy changed, 40% did not. Now, when we look at what was the implementation success across these 7 academic centers, again, we would argue that that was a successful implementation because in those who did not carry a loss of function allele, 85% were getting clopidogrel. So clearly more people were getting an alternative therapy in all of these centers if they had a loss of function allele, and the most common alternative drug was prasugrel. And so these are our data. These are presented as event curves rather than survival curves. But again, we showed basically exactly the same thing across these academic centers. So those who had loss of function, we recommended against clopidogrel, but got clopidogrel, had significantly higher rate of death, heart attack, and stroke than did those individuals who carried the loss of function allele and got alternative therapy. So that was significantly different. The non-loss of function carriers, who 85% of whom got clopidogrel, did similarly to those who got loss of function and alternative therapy. Again, not a randomized controlled trial, so this is really sort of a pragmatic implementation. And so we did propensity scoring to try to adjust for differences in the population that may have existed. And so the adjusted hazard ratio basically suggested that if you carry a loss of function allele and you are treated with clopidogrel, you have a more than 2-fold increased risk of having a death, heart attack, or stroke. And what I'll really highlight for you is most of those are in the first 1 to 2 months. For those of you, those who are sort of savvy to Medicare payment, Medicare will not reimburse a readmission in this particular scenario in the first 30 days. So there are also economic consequences, and there's really a strong economic story, and we're submitting an economic analysis in the next couple of weeks really around these data set that documents not only the outcomes benefits, but the economic benefits of a genotype-guided approach to antiplatelet therapy. So to summarize, we have now data in nearly 2,000 patients across 7 academic medical centers in the U.S. documenting that you can implement clinically pharmacogenetic testing, that physicians will change therapy based on the CYP2C19 genotype, and that the data really suggests that we improve clinical outcomes, specifically reduce death, stroke, and heart attack in these patients. Next, I want to talk about CYP2D6, another very important drug metabolizing enzyme that also has common variation and in particular common loss of function variation. So multiple alleles that can lead to a loss of function. There are also what we call reduced function alleles. This is also an interesting gene. Many have argued that CYP2D6 and HLA, the HLA complex, are sort of the most complex genes or gene regions in the human genome. So CYP2D6 is particularly interesting. Not only does it have all this loss of function type of variation, but it also is a gene that undergoes duplication or multiplication. So you can have 3, 4, 5. In the literature, the top is 17 copies of CYP2D6 allele. And so that can lead then to the ultra-metabolizer phenotype when you have duplication of functional alleles. So a similar process where we break down into intermediate, normal, intermediate, poor, or ultra-rapid. And again, not quite as frequent as the CYP2C19 variability, but still very common. So 5 to 10% of the population do not have the ability to metabolize drugs through CYP2D6. I'm one of those. And a similar percent have significantly reduced function, a smaller percent, but still certainly not uncommon, 1 to 2% of the population have this duplication of a functional allele that leads to the ultra-rapid metabolism phenotype. The other thing that's really important about CYP2D6 is it has very, very common and potent drug interactions. So many commonly used drugs that can essentially phenoconvert somebody into a poor metabolizer, basically completely wipe out metabolism via CYP2D6. So this is probably the enzyme, if you really want to pick an enzyme to focus on enzyme inhibitors and drug interactions, this is an important one to do that with. And so this is a cartoon of codeine, which is sort of an example again. So codeine also is a prodrug, is not active or minimally active as an analgesic as administered. It's inactivated by CYP3A4, and its really only bioactivation pathway is through CYP2D6, and it gets converted to morphine. So if you're a CYP2D6 poor metabolizer, and you don't have CYP2D6, you can't generate the active metabolite and you cannot generate the analgesic effect. The same is essentially true for Tramadol, which is among the top 20 most commonly prescribed drugs in this country. More questions about hydrocodone and oxycodone. Clearly CYP2D6 is involved in their metabolism, but there's much less clarity on exactly what role it plays, because hydrocodone and oxycodone as parent compounds do have active analgesic properties. And so there are CPIC guidelines again. This is, I think, a cartoon out of St. Jude and their implementation. And so basically if you follow from left to right, for the ultra-rapid metabolizers, the recommendation is to avoid codeine or Tramadol, because the ability is actually to generate excessive amounts. So at a, quote, normal dose, those individuals would generate an excessive amount of the analgesic. And there are data in the literature of death, particularly of children and infants, who either were ultra-rapid metabolizers, in one case a mother who was an ultra-rapid metabolizer breastfeeding a baby who died of respiratory depression due to excessive morphine exposure. So avoid because of excessive risk of toxicity in the ultra-rapid metabolizer. In the extensive metabolizer or normal metabolizer, it's fine. In the intermediate metabolizer, there are several ways to go. At St. Jude, I think they recommend using it, but with caution. In our institution, we elected to recommend against CYP2D6 and recommend against those drugs in intermediate metabolizers. And then in poor metabolizers, also to recommend against the use of tramadol or codeine because of the inability to activate to the active metabolite. And so we decided to implement, unlike we did with Clopidogrel, which really was truly just a clinical implementation and we collected the data through the electronic health record, we decided to do a pragmatic clinical trial. And so we enrolled 480 patients in our primary care setting at UF and some partner practices in Orlando. We randomized patients 2-to-1 to a genotype intervention versus usual care. It was actually a clinic-based intervention. So clinics were really 2-to-1. And so we had baseline data on pain, their pain intensity. These were patients who had at least 3 months of chronic pain. And so we collected patient-reported outcome using NIH-developed PROMIS measures. So on a scale of 1 to 10, the average pain of our patients was 6.7, so pretty clearly patients who were in chronic moderate pain. So this is sort of a cartoon describing what we did. So we had genotyping, which we did by a buccal swab, was sent to the pathology lab, and then the genotype results were reported into the EHR at about 7 days on average. Again, this is a little more complicated gene because you not only have to genotype SNPs, but also determine the copy number to figure out if there is the multiplication or duplication. The genotype was then reported back, and we had really a very high level of pharmacist intervention in this. And so we took the activity score based on the genotype and then superimposed on that the drug interaction. So for strong inhibitors, we multiplied the activity score by 0. So if they were on a strong inhibitor, then their predicted phenotype was poor metabolism. If they were on a moderate inhibitor, we multiplied their activity score by 0.5. And then based on the activity score, had a predicted phenotype, and based on the predicted phenotype, made a recommendation to the physician. So this was much more intensive, was not sort of an automated best practice advisory. We're trying to figure out how to automate that in our next program. And then, though, it was up to the physician whether or not they wanted to make any change in the pain therapy for the patient. So these are our data based on both the genetically driven phenotype and then once we encountered or included the drug interaction. So this is a chronic pain population, and so they had a lot of use of antidepressants, and antidepressants are sort of common in this list of inhibitors. So about 5% were poor metabolizers based on genotype. When we included drug interactions, it went up to 18%. Similar number for intermediate metabolizers again. That went way up when we included the drug interaction. So when we looked at drug interactions, it phenoconverted 22% of these patients to intermediate or poor metabolizers. So you can see over 32% of the patients in this cohort, based on genotype or drug interaction, were considered intermediate or poor metabolizers. And so in that scenario, we recommended against use of codeine and tramadol, and we had slightly softer cautions against hydrocodone and oxycodone. So these are our pain data. So just to orient you, these are pain scores. So the higher the score, the more pain. So if you reduce the score, that's good. And so what I'm showing you here, our primary hypothesis was that in intermediate and poor metabolizers, taking tramadol or codeine at baseline, if we recommend to change therapy to something that doesn't rely on CYP2D6, their pain score will improve because they'll be on a better analgesic therapy. And so on the left portion of the graph are the data for our intermediate and poor metabolizers in our genotype-guided arm versus our usual care arm. And so you can see that we showed a significant improvement in pain scores over a 3-month period for those in the genotype-guided arm. On the right-hand side are those who are normal metabolizers. And so we didn't recommend any drug therapy change. We wouldn't expect, in that case, the genotype to change anything. And indeed, the pain scores were essentially identical in the CYP2D6 guided. And so one of the questions we had when we first presented these data, how do you know this just isn't a placebo effect? Somebody knows their CYP2D6 genotype and they think their pain management is better, so it's not really better, they just think it's better. And so we really, argue that the data showing really identical pain scores in the normal metabolizers is our evidence, part of our evidence for a lack of a placebo effect, because they did know their genotype. Or had the ability to know their genotype. And so again, documenting that we can improve patient outcomes through a genotype-guided approach. For the FDA, if you want to get approved as a drug therapy for pain, you have to document a 30% reduction in pain scores. So we also looked at that. In our IMs and PMs, we had significantly more patients, 24%, who had a 30% reduction in pain compared to none of our patients in our usual care arm. In our normal metabolizers, there were no differences in the percent with a 30% reduction. The p-value for that was 0.95. So again, really evidence of improvement in the overall pain score, but also what many would argue is a clinically significant change in pain as well. So then we were interested, because the literature is a little more foggy, for hydrocodone and oxycodone. So we looked at hydrocodone alone, and we looked at oxycodone alone. When we looked at hydrocodone alone, it looked exactly like the codeine and tramadol data. And so then when we added in hydrocodone, again, a very commonly used drug in this country, the data really in some ways were even more compelling for significant improvement in pain score in the IM and PM group, but again, no differences in the normal metabolizers, which is what we would expect. However, we saw nothing. In fact, we saw that in the oxycodone group, they did significantly worse in the genotype-guided arm. So we're still trying to sort this out, but we feel like our data really suggests that CYP2D6 may not be important, and we perhaps should not recommend against oxycodone based on CYP2D6 genotype, whereas in our next implementations, we really are including hydrocodone in that group. So again, to summarize, we've documented in a completely different setting, a primary care setting versus an acute intervention setting, that you can implement pharmacogenetics clinically, that it has, again, the potential to improve clinical outcomes. We do know that for the physician to use it most effectively, it needs to be available to them when the patient is sitting in front of them. And so figuring out ways to make that happen is really an important challenge. So not always did the physician change the therapy in the first 3 months, because a lot of times they didn't see them again in the first 3 months, and they didn't necessarily call them up and change their drug therapy. When they did make changes, 95% of the time they followed our recommendation. We also believe that this suggests that inclusion of drug interactions is really important, and about 10% of our population was IM and PM based on genotype only, but we used drug interactions as well, and that was over 30% of the population. Just speaking more broadly about pharmacogenetic implementation, I think all of us believe that the best approach is to test multiple genes at once, have those available in the electronic health record to use them over time, because there are many very commonly used drugs that we can guide by pharmacogenetics. And so we looked in our electronic health records for UF Health, both in Gainesville and Jacksonville. So in somebody who was prescribed an opioid, in the following year, how many of them had another drug for which there was a guideline? And so we focused really on just chronic therapies. We didn't look at cancer drugs in this. And there's not a PPI guideline, but there's one in the works, so we included that. And so you can see that it's really very common. So 65% of patients in our health system who had an opioid in the following year had at least one more drug where they could have therapy guided by pharmacogenetics. And so we think that really argues for panel-based pharmacogenetic testing, get all of it in there at some point for patients, and then just have it available to guide their therapy. So what have we learned from our pharmacogenetic implementations? One is that physicians or prescribers will use genetic data to guide treatment decisions, but it needs to be available to be most effective. It needs to be available at the point of the patient-physician encounter. And so part of our challenges are working to figure out how to sort of overcome and achieve that. It's very clear that we just need to provide very clear guidance. They don't want to know the literature. They don't want to remember what's the genotype, what does that mean, what do I do? And so building very clear clinical decision support tools is really essential for this to work. We also know that patients are really enthusiastic. What I didn't mention to you is that in our usual care arm of that pain trial, there were two things. First of all, when we went to physicians, they were not willing to participate in the study if we didn't offer it to everybody. So if we weren't going to offer it to the patients in the usual care arm, they weren't willing to participate. More importantly, we offered to the patients at the end of the study, whether or not we collected the genetic sample at the beginning, but we held it, and we asked them at the end if they wanted their genotype. 95% wanted their genotype reported into the electronic health record. So patients are really, really enthusiastic about having this information to personalize their therapy. As I showed you, there are many chronic and commonly used medications that we can guide. And so again, focusing just on those with CPIC guidelines, a high percent of patients, at least in the primary care setting, have utility. And as I think both Pam and Linda mentioned, education of users is essential. And so I say users in a very broad sense. And so I'll just wrap up and talk a little bit about some of the things we're offering at UF. So we have a precision medicine conference. It's in March. So for those of you who live in this part of the country, our weather might be better in March than yours. So we would love to have you join us. We offer both CME and CE and CPE, and really are trying to focus not just on pharmacogenomics, but more broadly in genomic medicine and precision medicine. So our fourth conference, we don't have our date set. We should have our date set in the next couple weeks, but our fourth conference will be in 2019. We also will be launching this fall an online graduate certificate. So this is not a CE program. This is not a 20-hour CE program. This is a graduate certificate. So 9 credit hours of graduate coursework. It will be online and asynchronous, so people can sort of do it at their leisure in some ways, but a given course will be offered over a 5-week period or a half semester or a full semester. So they'll have assignments, but can do it as they want. And as I've mentioned, we'll be launching that this fall. And so we will have both required courses and elective courses. The required courses will include genomic technologies, molecular genetics, genetic epi, and then the precision medicine seminar, which is our conference, will be one of those. And then we'll have electives in precision health, precision public health, oncology, pharmacogenomics, and ethics and communication. So this is the website, and if you are interested or you know anybody's interested, I mean, we really believe that programs like this are great as introductory, but it's not a knowledge area that you can learn everything you need in 10 or 20 hours. And so we think that this is an important way to really help advance the field. And then I'll just mention we just got funded by NIGMS, or NHGRI. We're the only program based in the College of Pharmacy for a genomic medicine training program. So we just enrolled our first 2 trainees. We have a very strong focus in enrolling clinicians. We will enroll PhD trained individuals, but we're expected to enroll at least 60 to 70% of our trainees to have a clinical background. And so again, for those who are more junior in the audience and interested in really advancing your skill set, we'd love to talk to you. So to summarize and conclude, genotype-guided approaches can improve clinical outcomes. We have to continue to develop evidence, not just that there's an association between genotype and drug therapy, but that we can improve clinical outcomes. And we have to continue education. It's a very steep uphill climb if people don't know how to use this information. So I'll just acknowledge and thank the people that have been involved in some of the data I presented to you. It takes an army, as you know, and we've also been very fortunate to have substantial funding from NIH to conduct this work. So with that, I'll thank you, and hopefully I'm not completely over time.
Tato Hoshitsuki. Dean Jonathan, thank you for that excellent presentation on the clinical need for evidence and the clinical piece of it. My question is, you alluded to a few times the need for randomized controlled trials as that is a gold standard. And if you could speak a little bit about whether that's appropriate or inappropriate to have that as a gold standard for pharmacy.
Yeah, so this is a challenge. So, and particularly in cardiology, so we published the data I just presented. We published in JACC, Cardiovascular Interventions, which is pretty high impact. It's a primary journal that interventional cardiologists read. And we had a nice editorial that was written along with it, but the end of the editorial said, now we need a randomized controlled clinical trial. And you're like, really? Frankly, I'm not going to take Clopidogrel unless I know my CYP2C19 genotype, nor is anybody in my family going to take that drug. So, but there are certain communities, and cardiology is probably the worst, frankly, that says without randomized controlled clinical trial data, I'm going to ignore it. Now the reality is there is an RCT that's ongoing led by Mayo Clinic, but it has taken, I think it was supposed to be done several years ago, and the latest I heard was 2020, but I don't know, maybe it will be 2022. So the reality is that they're expensive, they take a long time. The FDA would not allow randomization of poor metabolizers because it's got a boxed warning, right? So you begin to get these ethical dilemmas with the IRB about whether ethically you can randomize patient, for example, randomize a poor metabolizer to Clopidogrel. And so I think our argument is that we need evidence, but probably randomized controlled trial evidence is not what we should try to focus on, and that we should try to implement more pragmatic trials, which is really what we did in the opioid trial, so that you're still having two separate groups. You can really see how the uptake is clinically, so you kind of answer two questions at once. So the Ignite Network, which I mentioned, will be renewing in the fall, and the focus is going to transition, and it will be focusing on conducting somewhere between 2 and 4 national level clinical trials, but they're all going to be pragmatic trials. They're not going to be RCTs. I think there's just, the reality is we can't answer every clinical question in the world with an RCT. But there are some clinical communities where that's a really, really hard argument. But I think, collectively the field is really trying to argue that pragmatic trials are the way to go.
Thank you.
Mm-hmm. I had a quick question about the 2C19 Clopidogrel. Were they given an opportunity to put an override reason or a reason to not? I would be curious to see what was sort of...
Yeah, I didn't mention that. So we had sort of 3 checkboxes that they could click out of that to get out of that BPA. The third one was to continue Clopidogrel. If they clicked that, then we popped up and asked them to tell us why. They didn't have to. They could click past that. The reasons that they continued Clopidogrel were multifold. We started our implementation in June of 2012, and in late May of 2012, Clopidogrel went off patent. And so the cost of Clopidogrel plummeted. So, at Walgreens or whatever, or Walmart, was like $4 a month. And the other two are about $200 a month, and even people with prescription benefits was often $50 to $70 a month. So cost of therapy was a very, very big reason that they would continue Clopidogrel. And there are recent data that have shown that use of antiplatelet therapy, as there's been more and more use of ticagrelor and prasugrel, has actually fallen, and it looks like it's really a cost-driven thing. So we always have to be aware, a drug with reduced efficacy that a patient takes is still better than a prescription they don't fill for a more effective drug. So there's always this balance. So that was probably the number one reason. Another reason really had to do with the contraindications and cautions. And so, if a person had, some clinical situation that made one of the other drugs tricky, then they would stay. And sometimes there's just, there was no real good reason. But those were the two. So the cost and, problems or concerns about the other drugs were probably the two most common.
Thank you. Pat?
Yeah,
oh,
and sorry, just one more
ahead.
question about the 2D6. You had mentioned the hydrocodone data was similar, and just to clarify, did you recommend against it in all the IMs and PMs?
So we did recommend against it, but it was slightly soft because, again, it was a consult note. It was slightly softer language than we used for tramadol and codeine. Yeah. Now, in our next intervention, we'll probably use slightly stronger language for hydrocodone.
Thank you.
Yeah.
Hi, Pat Gleason, Prime Therapeutics, PBM owned by Blues Plans. So I'm all about cost effectiveness. So when you mention, and I very much want to use Clopidogrel because it's generic, but if I have to spend for a test for everybody and then the drug cost versus Tegaglor, and I'm pronouncing it correctly, but Brilinta. So help me out from a cost effectiveness standpoint here. You didn't mention the cost of the test, so can you go into some detail there?
So, you know, the Medicare reimbursement rate is, I think, $280 for the test. And so this paper that we'll submit in the next couple weeks, we modeled all of those scenarios. We modeled Clopidogrel in everybody, no genetic test, Ticagrelor in everybody, no genetic test, genetic testing with Ticagrelor for the first month, then Clopidogrel in appropriate patients. So there are like 5 or 6 different scenarios that were modeled. And basically, genetic testing and appropriate drug therapy based on the genotype was superior from an economic perspective to all the other models. Ticagrelor in everybody was sort of second. So that was sort of the next most cost effective. But I mean, what we hear from a lot of physicians is Ticagrelor is tricky. A lot of patients have sort of this shortness of breath. It's really bothersome to them. The BID dosing is bothersome. So we actually think that, and we're working on perhaps putting together, a national trial. We think that the best scenario may be Ticagrelor in everybody for the first month. You can actually get the company to pay for the first month, and then genotype test. While they're in the cath lab, you have a month to get the result back, and then switch therapy to Clopidogrel if they're not an IM or PM. And we think that that really may be sort of the best way to go.
Did you use the AWP cost, or did you do net cost with rebate?
So the models did not include, the economic modeling did not include the rebate. Yeah.
And then, oh, sorry, there's another question. But quickly, what is the role of the pharmacist? I heard the physician is seeing it. I guess I'm looking for the interprofessional team and the role of the pharmacist there in helping the patient.
Yeah, so I mean, I think pharmacists have a huge role to play, and we've actually just started, and we just hired our second pharmacist for a clinic, a consult clinic. And so what we're finding is that now that physicians in the primary care setting have sort of seen this, they're referring what they're seeing as complicated patients to our consult clinic. And so I think pharmacists have multiple roles. One is sort of as the primary provider, if you will, in a pharmacogenetic consult sort of setting. Second is as that sort of side-by-side team member with the physician, sort of guiding them on what to do. But they also can play, I think, higher roles in terms of just implementing in the health system. I mean, we have one of our pharmacist informaticians has really been key to our clinical decision support. So if you looked at our org chart for our program, there are pharmacists really in pretty much every single possible role except the pathology lab.
I think we might be out of time. I apologize, Pam, but once again, want to express our appreciation for Julie and her insightful review of some key issues, and thank her for obviously making the trip up here during our good weather time. So thank you, Julie. Absolutely. Yeah. Thank you very much.